It's interesting that the age that we're living in now, people are very interested in what we call the genome, genes. The whole collection of genes is called the genome. And the reason is because every property, of human beings, every trait of human beings is basically determined by his genes. And the idea of I would say, research from the 1960s until today has been to try and understand this genome. And by understanding what's coded inside our genes, we would understand all of the traits of human beings. And that's been the center of attention of modern biology, modern genetics, modern molecular biology. How it all works, what's the machinery for translating the genetic information into the trait information, how does it work? This is the research from 1960 until today. And it turns out that one of the most important aspects of understanding man's behavior is not the book in which is written all the information, but rather how that book is controlled, how that book is read, how that book is regulated. Right? And that's where we come in. Right? And our work basically pioneered, first of all, in the concept that there's control, that the genetic information is basically important, but if you don't know how to read it, you don't know what to do with it, you don't know how to control it, what genes to turn on and what genes to turn off, it's useless. And we developed this subject, and we discovered one of the major mechanisms of how this is done, how genes are regulated. And it turns out that it's very important. It's very important for understanding human development. It's very important for understanding cancer. It's very important in agriculture. It's very important for in genetic manipulations, for people who want to do manipulations. And it's very important in diagnosis of diseases. It turns out to be what I would call maybe the second foundation of genetics. The first being the information itself, the second foundation is how it's controlled. So you can compare our genome, our genes, to a text. Mm-hmm. And we have within us, in fact we have in every cell of our body, this whole text that provides information for how to make the components of the body. And this text has to be read. And it's the reader is control of the reading. So you can imagine, for instance, a text, take a page of text. If you wanted to do that at the level of text, you would have all the words and the letters and words and sentences and paragraphs. And you might cross things out. You might make a comment over here. Right? You might circle some things. You might erase some things on the text. You might add things. That's the way we control how the text is read and understood. We call it a process of annotation. And the equivalent of that in the human body is what we call DNA methylation. DNA methylation is a modification of the text. In the body, the text is not a page, but rather it's a chemical molecule. It has letters and sentences just like any other text, but it's all chemical. Okay? So each letter is a different chemical. And what DNA methylation does is you can modify that text. So you can take part of the text and put on it a methyl group, a chemical group. And it doesn't change the text. The text is still there. Right? But when the the machinery comes along to read this text, it sees it as an annotation. And it says, oh, this area is methylated, I don't read this. Right? And so methylation serves as a way of turning genes off and not using them.